one of the key aspects that I would put squarely in this area is the involvement of both regulatory and medical affairs at a very early point in drug development. And I make the distinction between, here's a drug and here's a product. Our desire would be to make products that are effective, that are helpful, that are meaningful in terms of patients and conditions or treatment of patients. But really understanding how to link the science to the application of the drug and where it fills the gap in what is necessary is important in the design of your drug and the design of your drug's development process.
What does it take to bring a life-saving therapy from a research idea all the way to a patient's bedside? Few people have stood at more points along that path than Milan Tomic. He has been 30 years in biotechnology. He developed 5 antibody co-mixture products from DNA to phase 1 and worked on a 2,000-liter manufacturing facility and brought that up to EU IMP certification. Today, he is building Albrem, his own company. In part 1, we explore the career decisions, the pivots, and hard-won lessons that shape that journey. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Milan, to the show. It's good to have you on today.
Thank you. I'm glad to be here. I'm looking forward to this chat.
To get us going, Milan, let me ask you this perhaps controversial question. Share something that you believe about bioprocess development that most people disagree with.
It's a very good question, and one that I had to think back through what I have done and really where I am now and how I approach things. I think one of the key aspects that I would put squarely in this area is the involvement of both regulatory and medical affairs at a very early point in drug development. And this puts you really at a point post-discovery when you know some of the basics of the drugs, looks like it could work, getting an idea of really how this can be affected during the translation of your drug into an actual product is very critical. And I make the distinction between, here's a drug and here's a product. Our desire would be to make products that are effective, that are helpful, that are meaningful in terms of patients and conditions or treatment of patients. But really understanding how to link the science to the application of the drug and where it fills the gap in what is necessary is important in the design of your drug and the design of your drug's development process. It's one of the key aspects, I believe. What I have seen is that a lot of people put regulatory into the compliance section, into quality control or such. I do believe that both regulatory and medical affairs belong more in development, more in the strategy. of developing your drug for a market.
Milan, you've got an exciting career story, an exciting journey. Take us back to the beginning. What first drew you into molecular biology and biochemistry, and what were some choices that you took from the bench into increasingly senior operational and leadership roles?
It's not quite a random walk, but it is sort of, I think, if you want a catchphrase for it, connect the dots kind of exploration. I really developed a curiosity of how things worked and then pursued that. And if you look at it that way, not always will you have your own path in a career or your own selection of things to do, but there are lots of things that are interesting in the overall direction that you would like to grow your scientific understanding or even your career and your personal advancement in. And so the path in this case was more, I studied science. I became interested in development. I became interested in gene regulation. That led to more of graduate work in actual protein DNA interactions, the actual molecular interactions. And so I really began to have an interest in that aspect of it. That actually led to my first role in terms of structurally modifying a protein at my first job in industry. And that, of course, led to, there are more questions and more curiosity about how a drug is actually designed or developed, and how do you do that well, and how do you do it correctly was really a curiosity that drove my varied interests. Surprisingly, and I think not maybe even easily discernible, at least in the beginning, was that trying to figure out how things are connected leads to advancement because you have cross-fertilization, as a lot of people like to use, and you begin to see where there are inefficiencies between different steps of drug development or process, if you want to call it general sense, that can be linked. And these improved ways for any other description result in, there must be something good about what you're doing, and that results in your career advancement. And that results with you having more opportunity to do the same and have, if you will, more growth to connect the dots and develop what you're interested in. And that really is something that I would say at the root of it exists, is that you have to be interested in what you're doing. It'll drive a lot of your enjoyment of your work.
Is that curiosity about how things work, is this also what led you then to leave traditional leadership role to step into a founder's role, or how did that come about?
I think one of the important aspects, your job is your job, your work is your work. And as much as I can say that you should enjoy your work, you should have fun at your work, which I always strive for, there's also a family aspect. There is also a life aspect. And you have to be able to, at some level, integrate those. So what could drive your job or choice or even position is really how does your family integrate with your work and interest. And in my case, it's not just family, it's location. Are you comfortable where you are? Disliking the fact and really hating the fact that you have to drive in snow for half the year is not going to be helpful for your enjoyment in your career. Not liking humidity or not liking cold or heat, those are important factors. But in any of these, choosing how you move forward is kind of a mix between what you think should be done and how that can be achieved. My curiosity, my interest in linking things, connecting the dots is actually what led me to this role. And I explored different aspects during my industry careers, and now it came to the point where I could actually look at how some of these aspects can be integrated. And that wasn't as easy for me to find. So I wanted to do that kind of investigation, that kind of curiosity, that kind of furthering my interest, rather than go into larger management roles where my focus would be on other things, managing people or having less science in my role. I hope that answers the question.
What was the most surprising part as you were stepping in to the founder role at Albrem, your own company? What were some unexpected things that happened, and what do you like about this role now?
Well, the answer would be different for different people. I think what I like about the role is what I just described in the previous question was that it allows for an integration of the kinds of concepts that I feel are important in the drug development area, and that is huge to me. What I really didn't expect was that it requires a lot of salesmanship. Business I can understand. Business I have no problem with. But salesmanship, in my mind, doesn't dive deep into the how of things. It is a very different skill that a lot of, say, highly analytical people, scientists, may not have experienced. And so for me, that has been a real, if you will, wake-up call that, wow, I can't go as deep as I'd want into every situation. I have to focus on getting the business off the ground. And that is a different responsibility driver that caused me to now work more on my salesmanship.
This is so accurate. Like you, for me, running my own company was one of the biggest aha moments. Sales and selling, these are everywhere. It's just all about selling.
It is. It's pervasive in almost every part of it. And my own experience was really let the science, the data speak for itself. Others should recognize it, and that's not a part of sales. Sales is really informing in a larger sense. That is not always as easy for someone who wanted to always look at the how. That was a wake-up moment.
Let's unpack different parts of science. You have worked on various parts of science and various areas of biotechnology. So let's go back to your postdoc work. Tell us a bit, what was the focus and what did you learn during that time about science? You alluded a bit to therapy, translating therapy into what really works, but tell us a bit more. What were specific learnings during your postdoc?
It is a difficult thing to say there were specific learnings, at least in my case. I mean, I've known people who were very directed and wanted to focus their, I'd say, career path very, very narrowly. And that wasn't so much my interest. My interest was finding generally how things worked and how they were connected. And so when I started, and I'll go back a few years before my postdoc, when I started my work as a graduate student, I actually did want to study the interactions of DNA and proteins. And the graduate work for at least studying these things really did let me into that area, but we got scooped. And 3 years of my graduate work I couldn't publish. Now, you could take a look at that as a real crisis, but it also led me to kind of piece apart what I was doing and say, okay, well, if I can't do both, if I can't look at, for example, protein DNA recognition and binding, I could look at DNA by itself. So I looked at the different structural elements on its own. Graduate work was in nucleic acids and nucleic acid structure, and that led to a controversial thing that my PI was pushing, and that was good. I mean, we were able to show that those kinds of things did make a difference in the flexibility of nucleic acids, but then nucleic acids were nucleic acids. There were 4 repeats. Proteins were much more interesting, much more dynamic. And I got the opportunity to study the protein structure in my postdoc. And in a way, my postdoc really was looking at that recognition molecular interaction in a piecemeal way. And after that, the work in proteins, the work in analytics led to my first industry job, which fortunately for me was also in a structural protein field. What I'm trying to say, it isn't always, oh, I'm going to go and get this kind of work. It's more, in my case, that curiosity, desire for deeper understanding allowed me to investigate different aspects that were also of an interest to me. So I didn't lock myself into just studying proteins or just studying nucleic acids. It was more, gee, let's find out about the how of this or that. They can all be very interesting.
Yeah, it seems to me that your curiosity was driving you to go from one thing to another, and then yet another thing, and then you had another question which led you to another area of science. And I think this curiosity also led you then to what seems quite a different field, where you were involved in the design and upgrade of a 2,000-liter manufacturing facility. Tell us more about that. What happened then?
The interesting thing then was really something where you sort of have to look at that point that you brought up, which in my career has really resurfaced multiple times, and that's curiosity, finding out about the how of things. And when I moved from Bayer, which was more focused on Factor 8, I moved more into quality control because I was interested in how the quality aspects of the process were figuring into and driving really the evolution of a drug or a molecule towards being a drug, and wanted to learn more about what is involved in quality. And prior to that particular project or work that you're describing, I had the opportunity to work in QC, worked on efficiency, worked on product release, raw materials, as well as protein product, had a good understanding of some of the nuances that were involved in part of the process of drug development. And some of these were good. There were some that honestly I really thought were good molecules, but failed for legal or liability potential reasons. And that was interesting. Some failed because the whole process and design was very, if you will, specific. And they really didn't find a good niche. The molecule just wasn't as active as it could have been. And that was a bacterioporin indicator that Zoma was working on at the time and required, honestly, putting resin beads in a bioreactor, which required different impeller speeds and design. So you had a lot of process knowledge and learning while I was in quality. And that led to quality engineering, validation, and some quality assurance type functions as well. But the plant itself wasn't efficient. And as we were entering into that stage, it seemed like there weren't any people that could take on that role. And so I kind of brought it up for management that this can be improved. This really can be something that you can make better, that you can have a better, stronger link, a more efficient design. And that kind of landed me into the job of rebuilding the plant. And since I had been in quality, had the aspects of really what kind of workflows seemed to be most efficient, and had in mind that you needed to have good throughput, I had an opportunity to apply this, figure out the how, and see if I could essentially use the knowledge I had gained in quality to improve the workflows and the actual facility itself. And that was very interesting. It led me into a lot of understanding of the plant and how a plant itself should be designed. And that was a great learning experience. I'm glad it was so successful for Zoma as well.
From this extensive experience and probably sometimes also challenging experience as you were learning from past failures and then redesigning the factory, And also, since you have later worked on bringing products into phase 1 and phase 2, what are some pieces of advice you can give to a smart biotech scientist listening? Because some are working in smaller companies and need to figure out CMC. Perhaps others are in bigger companies, but CMC is challenging. What is, from your perspective, the most important aspects people should watch out for?
From a sense of a product development, that's assuming that you've discovered the right molecule. I think the biggest thing to watch out for is the application of that product itself and the actual production of the product. So during development, you have choices in how you design a process. Understanding how to make that process robust and how to really make it fit into a particular plant. And by plant, I mean something that would allow you to produce consistently GMP materials is important. And you need to take that into the design of the molecule, into the design of the development, as much as you need to take into account the actual targeting of the drug itself. So those two working together will give you an idea of scale, process that can be really implemented, and how your drug fits into the whole thing. That, from a new starting scientist who has a great molecule and a good idea, will give you a good position to be able to talk with your investors, talk with your partners or founders or other collaborators in a way that you can show that it's not just, oh, I've got something. It's more that I've got something and it can go ahead and advance and work to fill a gap that's necessary. And that will give a lot more traction to your advancement.
From a Berkeley biochemistry lab to the operational complexity of a 2,000-liter GMP facility, Milan Tomic's trajectory is a masterclass in how scientific depth and operational discipline compound over time. In part 2, we will shift from biography to practice. We'll look into the valley of death, cell-free protein synthesis, and what it actually takes to move a promising protein candidate toward the clinic. If this episode has been valuable, please leave a review on Apple Podcasts or your preferred platform. And thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.